This presentation is about a research of effect of Lactobacillus plantarum KFY02 isolated from naturally fermented yogurt on the weight loss in mice with high-fat diet-induced obesity via PPAR-alpha gamma signaling pathway. There are three major discoveries that were found in this research. The first one is the researchers found that Lactobacillus plantarum KFY02 significantly reduced body weight and fat accumulation in mice fat with a high-fat diet. This indicates that this probiotic has strong anti-obesity properties. Additionally, the administration of Lactobacillus plantarum KFY02 also improved lipid metabolism. This was evidenced by better lipid profiles including lower levels of serum triglyceride, total cholesterol and also LDL cholesterol. Lastly, the beneficial effects on weight and lipid metabolism were linked to the activation of the PPAR-alpha gamma signaling pathway. This pathway is important in regulating lipid metabolism and adipogenesis. The study hypothesized that Lactobacillus plantarum KFY02 strain could mitigate the obesity in mice that given with high-fat diet by modulating the PPAR-alpha gamma signaling pathway.
The objective for the research article is to study the weight loss effect of LP-KFY02 in obese mice. Next, to investigate the role of the PPAR-alpha gamma pathway in LP-KFY02's action on lipid metabolism. Lastly, to compare LP-KFY02's performance against other treatments for obesity.
In this study, major methods included animal experiment, biochemical analysis, molecular biological detection, pathological observation, and Western blot analysis. Animal experiment involved 60 mice divided into groups fed either a normal diet or a high-fat diet with various treatment. Biochemical analysis assessed serum and liver biochemistry to evaluate lipid profile. Molecular biological detection used qPCR to examine gene expression related to lipid metabolism and inflammation. Pathological observation evaluated tissue changes using H&E staining to assess tissue damage and fat accumulation. Western blot analysis analyzed protein expression levels to understand effect on lipid metabolism.
In a study on mice, those fed a high-fat diet gained weight significantly faster than those on a normal diet. However, mice treated with L-carnitine, LKFY02, HKFY02, and LDSP had noticeably lower body weights compared to the high-fat diet group. This suggests that the high concentration of LP-KFY02 can effectively prevent the weight gain, indicating its potential in obesity prevention. In a study on liver tissue, normal mice had clear and healthy liver structures with no fat buildup. Mice on a high-fat diet show significant liver steatosis and fat accumulations around the blood vessel. Treatment with LP-KFY02 significantly reduced these issues, suggesting it can decrease the liver fat accumulations and also help to maintain the liver cell integrity, potentially improving liver functions affected by a high-fat diet.
Here are a few considerations for alternative interpretations of the study's result on Lactobacillus plantarum KFY02 and its effect on obesity in mice. First, dose-response relationship. The study found that LP-KFY02 induced weight gain in mice. However, the interpretation could be expanded to consider the dose-response relationship. If the study only tested on one dose of LP-KFY02, it will be valuable to know if higher dose or lower dose will have a more significant or diminished effect. The second one is long-term effects. The study provides a result from an 8-week experiment. An alternative interpretation could consider the long-term effect of LP-KFY02 on obesity. It will be important to know if the beneficial effect persists or if there is a point at which the body adapts and the effect diminishes.
The research concludes that Lactobacillus plantarum KFY02 shows promise in preventing weight gain in high-fat diet-induced obese mice by influencing the PPAR-alpha gamma signaling pathway. LP-KFY02 exhibits benefits similar to L-carnitine and superior to Lactobacillus delbrueckii subspecies bulgaricus in lipid metabolism, organ index, and liver tissue damage elevations. I agree with the author's conclusion because LP-KFY02 significantly reduced organ index and lipid indicators in serum and liver, improved HDL-C levels, and alleviated liver damage and fat cell expansions. Molecular analysis demonstrate LP-KF02's ability to regulate lipid metabolism genes, promoting lipid breakdown, and reducing genes associated with obesity and fat storage. Furthermore, LP-KFY02 modulated inflammatory response by reducing pro-inflammatory factors and increasing anti-inflammatory factors. Overall, LP-KFY02 showed potential as a probiotic for managing obesity and related metabolic disorder, offering a natural alternative to chemical slimming drugs.
Okay, so next is the conflicting and supporting evidence by authors. First is the conflicting. Due to the genetic variations from the mice, there are genetic factors that affect the effectiveness of LP-KFY02, which leads to the variation in related to the lipid metabolism and obesity management, which could result in conflicting evidence from different studies. While the supporting, there is evidence that LP-KFY02 have the ability to effectively reduce the body weight, serum cholesterol, LDL-C, and triglyceride levels in mice fed a high-fat diet.
One of the key limitations of these studies is species specificity. The research was conducted on mice, which might not fully replicate the human physiological response. Mice and humans have different metabolic rates, gut microbiota composition, and response to dietary interventions. Next is the study duration and the specific dosage of Lactobacillus plantarum used in this experiment may not directly translate to human scenarios. And lastly, we need to address the issues of controlled variables. Several external factors such as environmental condition, the genetic background of the mice, and the variation in the high-fat diet composition given to the mice can influence the study's outcome.